Hello, welcome to the Enzymes podcast. I'm going to go over what you need to know for the test. Going to go over enzyme regulation and what activation energy is. So you got to know those three things to be ready for our quiz. If you're watching this today, probably tomorrow. So our need to know slide, this is kind of your study guide for this unit. It's a really small unit, so if you're able to answer all of these questions, you know more than you need to know for the test, okay? We want to know the structure of an enzyme, what an active site is. The active site is the business end of your enzyme. If your enzyme is a Pac-Man, it is the mouth of that Pac-Man. It's the part that actually carries out the chemical reaction. A substrate is the reactant that binds to your active site. So anything that binds to an enzyme is a substrate. Factors that regulate an enzyme are pH, temperature, and salt concentration. pH is pretty straightforward. If it's too outside of the bounds, then it's not going to work. Same thing with salt concentration. If it's too far outside of what your enzyme wants, your enzyme's going to break. Temperature we're going to talk a little bit more about when we get to there, but that one's a little bit more complicated, just a little bit. And then lastly, the function of our enzymes. They speed up reactions, they lower activation energy, and I want you to know what that looks like on a graph, okay? All right, so first we have to define chemical reactions. A chemical reaction is any set of chemicals going from one set to another. So one set of chemicals moving to another set of chemicals. What we see in a chemical reaction is bonds being broken, bonds being formed, or a combination of the two, and bonds being rearranged. So why do we carry out chemical reactions? We got to stay alive. All the things in our bodies that keep us alive are chemical reactions, and a lot of them need ATP. So a reason plants and animals either need to eat or get energy from the sun is so they can make ATP to carry out those chemical reactions to stay alive. So kind of linking it back to our ecology unit there for a second. The reason we need to eat each other is we got to get that energy to carry out chemical reactions to stay alive. All right, so activation energy is really the key to understanding how enzymes help us at all. So activation energy is the energy needed to start a reaction. If to get the ball rolling, you got to kick a little bit first. So if you look at this little diagram here on the right, you'll see reactants having to jump up and over this little barrier before they can go down to the product side. On your diagram here, the level of the floor for each side, so for reactant and products, and up here for the activation energy, are all levels of energy that you need. So reactants start at, say, let's call it a energy 3, right? We'll call it 1, 2, level 3 energy. Our activation energy, let's say we'll call it 5. There's no units on these things, we're just calling it 5. So in order to get all the way down to level 0, our reactants got to jump up to a level 5, and then they can fall all the way down to the products, okay? So unless you have an energy level of 5, this reaction won't occur. So this is telling us pretty much the same thing, just now in graph format. But there's two types of reactions that this occurs with. The first type is called an endergonic reaction, where we're taking reactants down here at level 0, and they're becoming products up here at level 3. They have to, even though they're absorbing energy, they still have to go over this activation energy bump right there. We'll call this let's say level 4. So we started reactants with 0. In order to get to their product energy level, they have to go above it and then come back down. Same thing on your exothermic reaction or exergonic reaction is we have energy being released from a system. So reactants start at level 3. They want to go to level 0. But to get there, they have to go all the way up to a level 5 to get there. And again, these are random numbers. They don't have units on them. I just want you to get the idea. So if you can see from both of these reactions, you have to go over your activation energy barrier before you can get to the products. The reason that's so important is otherwise these reactions would happen spontaneously, especially the exergonic reaction here. If we were able to just roll from reactants to products, nothing would stay stable and these reactions would happen on their own without any kind of energy input. which for us would be really bad. We would walk around and suddenly catch on fire. That's not really what we want to do. And so we're pretty lucky that this activation energy barrier is there stopping everything from happening. Endo and exo. All right, so why are enzymes important to living things? We're going to answer that on the next slide here. Semi-chemical reactions are just too slow to actually happen in real life. If we didn't have enzymes to help us out with that, we would suffocate waiting for oxygen to hop off our blood cells. That just doesn't happen fast enough. So in order to speed things up and get them going at a living speed, we use enzymes to help that along. Another problem that enzymes help out with are lowering down activation energy. Some reactions would require way too much energy input to have them happen inside of living cells. They'd suck up so much of our energy, we wouldn't have enough energy for anything else. And so enzymes help lower that. So problem number one that enzymes are helping us solve is speed. Problem number two is lowering activation energy. Now you see that weird E of A type thing right there? This little a is called a subscript, and this capital E just stands for energy. So if you were to say that as it kind of translates out of chemistry terms, this would be energy of activation, which is the same thing as activation energy. Oh, there's the bell. So this is where we ended during our first set of notes. So if you want to either draw a line under where we stopped, or maybe just take a break at this point, this is as far as we got during our first lecture period on Monday. All right, and so this slide still ties into what we talked about Monday, but we just started this fresh on Wednesday. Is now we're looking at what enzymes actually do. Remember we have our energy of activation barrier diagram here. So little jumpy beans can't get over unless they have super high amounts of energy. So what an enzyme does is it takes that little wall and it crushes it down. It doesn't delete it, it is still there, but it pushes it down so now more of these reactions can hop over to the product side with less energy. Because it takes less energy, more of them are able to hop over, and thus it increases the speed. If we got more reactants becoming products, we're getting more products per second, our speed's gone up. So this graph is now showing us the same thing, but kind of in a more scientific way. Scientists don't really like to use cartoons and diagrams. They call it unprofessional or something. So they'll use graphs instead. And this graph is really showing you the same thing. In red right here, this little red arrow, we have with an enzyme. When we have an enzyme here, our activation energy is lower, just like it was in our other diagram. In blue, we have something without an enzyme. And if there's not an enzyme, then you have to go over the whole activation energy barrier before you can get down to the product. But when you have an enzyme there, you just have a little bump to go over, and it's not too bad, and our cells can handle that. All right, so now we're going to zoom in a little bit more on exactly what enzymes do. We know that they lower activation energy, and we know they speed up reactions, but now we have to figure out how they do that. We've talked about how they help us. Now we look at the more specifics as to why. So first, enzymes are really, really specific. Just like all other molecules, these ones are heavily defined by their shape. If they're not the right shape, they're not going to work. Because that shape is so specific, they only catalyze one reaction at a time. Remember, catalyzing just means to facilitate or make easier. I'll put it in the simplest terms here. It means that it helps. So kind. But they only help one reaction at a time because they are so specific. So because they're so specific, we usually name them after what they help along. So we'll take their substrate, or the thing that binds to it, remove the suffix that it has, and add ase to the end. So in general in science, if you see any word that ends with ase, that means it's an enzyme. There are a few exceptions to that, unfortunately, but for the most part, all enzymes end with ase. Okay? So for example, amylase, which is a sugar that we find in starch, or in flour, the starch that we find in flour, it's broken down by amylose. Or, excuse me, I said that backwards. Amylose is a sugar that we find in starch, the starch we find in flour, and amylase is the enzyme that helps break that down. Sorry, I haven't had my coffee yet this morning. That's a mistake. So if you want to write out how these reactions take place or what they look like without doing the whole fancy chemical structure thing, you can do it with words like this. So you have your substrate here, and you're going to write that on the left. Ooh, writing with a mouth is not so good in the morning. Then you draw an arrow, and either on top of that arrow or inside of it, you write the enzyme. So that means amylase is helping amylose turn into the product. And then our product is over here. In this case, it's lots of glucose. Because remember, if you break down a polysaccharide, you get monosaccharides out of that. So you don't need to write this slide down. This is just kind of saying the same thing, but now with a chemical structure. This is actually the enzyme you read about in your article. You had sucrase, which was taking your substrate, sucrose. OSE means sugar. ASE means enzyme. And it took your disaccharide sucrose, and it broke that bond in the middle. When it broke that bond, now you have glucose on one side and a fructose on the other side. That process is done through something called hydrolysis, which means you break apart water to get to break some bond. Hydro means water, lysis means to break. For example, sucrase breaks down sucrose into table sugar. All right, so now we got to talk about that middle process. The middle process is when our substrate and our enzyme are all bound together in what's called an enzyme substrate complex. So step one, our substrates float in and they bind to our active site. Oops. All right, so step one, substrate binds. Once your substrate's bound, sorry about that, the announcements came on. Step two is once our substrate has bound, we have formed the enzyme substrate complex, which I'm just going to call the ESC. Okay, step three, reaction occurs, and finally step four, product floats away. Alright, so you can either write the thing that's in type on the left or just write down what I have in blue. But essentially step one is your substrate binds to your enzyme. It binds to the active site. It forms the enzyme substrate complex, which is this thing right here. And then finally our product leaves. Now the important part is that right after step four happens, step one happens all over again. This is a really cyclic process, and it happens very, very quickly. Now to help you remember this, I'm going to give you a little analogy to help kind of figure it out. Now a lot of people have those friends that just love to play matchmaker with the people they know. They say two people and you're like, you need to date. I've decided this. So your matchmaker friend is much like your enzyme. And it sees two people, two substrates, that it says, yep, you need to be together. This is going to work. So they pull them aside and they form this enzyme substrate complex. And the three of them are talking. And matchmaker friend says, you know, you two are really great for each other. This just needs to happen. And so little substrate people who originally were too scared and couldn't get over their fear or activation energy, and now with the enzyme present, they say, okay, yeah, I think this can work. I'm brave enough now. I have someone else here that thinks this will work. Let's do it. And so they form a relationship, and that relationship is the product. Now the product floats away, and matchmaker friend is satisfied for maybe half a second, and then they decide, oh, I see another two friends, and they want to pair them together up too. And so they go back and they find more substrates to bind together. They form an enzyme substrate complex. They form a relationship or a product, and that product floats away. Okay? Now, some of these reactions need a little bit of energy to get going. Remember in the beginning, we had our lowered activation energy in red, but there was still a little bit of it there. Because that bump's not completely flat, we do need some kind of energy to help us get over it. That's where ATP comes in. All the power in your body is ATP. So ATP will come and it will bind to your enzyme to help you get over that little bump in the road. Okay? That's all you really need to know. ATP provides energy for reaction. In your analogy, that would be your matchmaker friend needing a rock star before lunch, before they have all the courage and the charisma to really start a relationship. Okay, don't worry about these words. There's a lot of them. I'm not going to test you on lock and key because it's an outdated theory. So all this is saying is that the enzyme substrate complex remains together until the products are created. So ESC, or the enzyme substrate complex, lasts until the product floats away. And this is the same thing we've been talking about, but now it's showing it in a cycle. So step 1 is right here, our substrate binds. Step 2 is here. We left out a step 2, we're going to add one step in the middle. The new step 2 is ATP binds. Step 1 is substrate. Step 3, enzyme substrate complex is formed. Four, reaction occurs. And 5, product floats away. If you're confused looking at your notes, like Mr. Hutton, we only had 4 steps in class. In class, we just combined steps 3 and 4. Okay? So when you form the enzyme substrate complex, that's when the reaction occurs. And in class, I just combine those to save time a little bit. But since we're on that wonderful land known as the internet, we have lots of time. Okay? So there's your 5 steps. If it makes you more comfortable, go ahead and combine steps 3 and 4. Okay? They mean the same thing. Or they occur at the same time. All right, now we're getting into a little bit more of regulation. So enzyme regulation tells you how fast that cycle from the slide before is going to happen. When enzymes working at optimal conditions, some enzymes can complete that cycle up to 1,000 times a second, which is pretty fast. But they only can carry it out 1,000 times a second if they have optimal conditions. So the 3 conditions we really care about are temperature, pH, and salt concentrations. We're going to talk about pH and temperature the most. Salt concentrations do play a factor, but we're not going to really look at those as much, as they do kind of the same thing as pH. So every enzyme has their own set of very specific conditions that they want. That's really important. Conditions are specific to your enzyme. Okay? Super important. Every single one will have a different set of optimal conditions. So for example, pepsin, which is one of those annoying enzymes that doesn't follow the ASE rule, sorry about that, pepsin lives in our stomach. And our stomach's a very acidic place, so we got lots of stomach acid in there. So pepsin's been designed for that, and so it works optimally around pH 2.5. Right? About right there. If you go too far outside of pH 2.5, like if you go down to pH 0, or you go up to, let's say, pH 5, this enzyme will break and it won't work anymore. As you can see on the y-axis here, we got enzyme activity. So this is the number of cycles it's able to complete a second. How many times it can go from 1 through 5 per second. If you don't know what those are, maybe go back a slide and read through those again. You'll need to know those 5 steps. So it's able to complete those really quickly if it's where it needs to be, and not so much if it isn't. Now trypsin, another enzyme, works at a different set of pH values. It looks like it works around pH 8 is where it wants to live. If you went all the way down to pH 2 and threw trypsin in where pepsin lives, then trypsin would break and it wouldn't work at all. So every enzyme has their own unique set of specific conditions. And this one's just the one for pH right now. There should be a title on this graph. Okay. All right, last slide. We're almost there. So the last one we're going to talk about is temperature. Remember I said we're not going to talk about salt too much. It doesn't really matter as much. It just means there's too much positive charge around and it messes up your enzymes because magnets. But temperature is a little bit weird. To understand temperature, you have to know that all heat really is the movement of little molecules. The faster they're moving, the higher the temperature. The slower they're moving, the colder the temperature. Okay? If you need to, say it over to yourself 3 or 4 times. The faster molecules are moving, the higher the temperature. The slower they're moving, the lower the temperature. So the way our substrates bump into our active sites is by pure, perfect random chance. There's some molecule just kind of floating around out in the world, not really sure where it's going, doesn't really have an agenda, it's just kind of floating around. If it's floating around really quickly, then it's really likely to bump into our enzyme and react. If it's going too slowly, though, our molecules are less likely to bump into the enzyme, and so it lowers our enzyme activity. So I'm going to say that one more time. The slower molecules are moving, meaning the colder it is, the less likely it is that the substrate will bump into an enzyme. All right? So as we raise the temperature, our enzyme activity starts to go up. T for temperature, E for enzyme activity. This is a big EA, so enzyme activity, not activation energy, okay? So as we start to go up, our temperature increases, our enzymes are getting happier. Oops. Try that again, that didn't go too well. Okay. Enzyme activity starts to go up. So it starts to go up and up and up until we reach some point at temperature when molecules are moving so quickly that the ones inside of our enzyme start to move a little quickly too. So enzymes, as you can see from this little picture up here, are folded in a really, really specific way. We keep them in that specific shape by using different types of bonds. We got hydrogen, a couple ionic bonds, and it's something called a disulfide bond that we're not going to worry about. Just know it's some other type of bonds. It involves sulfur. And so if we start moving too quickly, then these bonds start to break. If those bonds start to break, our enzyme will go from this pretty weird little S-shapey thing to a straight line. If that happens, our enzyme is now denatured. That's a super important word. I gotta see it on your quiz. If I ask you what happens to an enzyme when you heat it up, it denatures. So if an enzyme denatures, now our active site isn't there anymore. If our active site isn't there, the whole thing doesn't work. So as we increase the temperature, if we start from the very, very coldest we can go, molecules start to speed up, our enzymes bumping into more of them, it's having a good time. But then all of a sudden, our enzyme starts to become unstable. As it becomes unstable, it breaks, and all of a sudden it'll just drop off. So in this case, our denaturing spot is about where that little tiny X is. So we like to heat up our enzymes until they can't handle it anymore, and then they will break and they won't work anymore. Okay? So that's as far as we got in class, so I'm going to go ahead and stop here. If you're confused as to what to study, go back and check that first slide that says the need to know slide. Those are where we're going to be pulling questions from for Friday's quiz. All right. Good luck studying. Thanks for watching. Best of luck.